All right, thank you so much for the invitation. I'm really excited to be here in person. I know that I give a talk here virtually during the Zoom time, and I made sure that this talk kind of does not overlap that much with that talk. So by way of introduction, this is the group that essentially has done most of the work with trainees, and I'm going to be presenting the results today. And over the past 5-10 years, my group has been focusing mainly on three main areas. One is genomic risk prediction that deals with polygenic risk scores uncertainty, and this is the topic that I covered during the pandemic when I gave my lecture to you guys. Hopefully some of you listened to it. The other focus area of my group has been in methods for underrepresented populations, and I'll talk more about it today about this. And who knows, maybe if I'll come back in the future, maybe I'll talk about the third aim there, which is all about methods for functional genomics, such as transcriptomoid association studies and fine mapping and so on and so forth, which I know that you guys have amazing expertise in that area. It was really exciting to talk to you today. All right, so without further ado, I'm going to spend some time introducing admixed populations, and this is an area that it has a lot of conceptions and misconceptions. So I'm going to go slower in terms of deliberate in introducing the concepts that I'm going to be using today. And I'm really curious how you guys, see this, and I'm really curious to hear your feedback about all the things that we're doing. Then I'm going to dive into statistical methodologies that we and others have been developing to do what is called disease mapping. So this will be association mapping all the way to trying to understand how genetic risk gets propagated in admixed populations. Then finally with a very interesting topic that kind of like we started this year, and we're very, very excited and fascinated about this fundamental question that we're all grappled with, which is whether the genetics plays the same role on disease across different ancestry populations. And I'm going to try to argue that looking at admixed individuals or admixed populations is a unique way where we could answer this question. And finally, I'm going to show you a little bit of the results of how we are kind of operationalizing all these methodologies within our biobank at UCLA, which is called Atlas, and try to convince you that we should move away from clustering individuals into units of ancestry, and we should start thinking about a continuum of genetic ancestries. And again, this is a buzzword, continuum. I'll try to convince you that it's actually something that we can compute, and that's something interesting. All right, so when we think about admixture, generally we start thinking about admixture between different continental ancestry sources. So we typically think about African Americans, we think about Hispanic Latino Americans, which are a result of the transatlantic slave trade and other forced demographies or migrations. But in general, admixture occurs at all sorts of different timescales, all the way from Neanderthal introgression into the ancestors of current-day Europeans and Asians, all the way to current-day, mixture or admixture of different genetic ancestries within, virtually every city worldwide. Well, people kind of having ancestries from various different genetic sources. And really what we're starting to emerge to see in the biobanks is that mixture between different subcontinental ancestry is ubiquitous, and virtually everyone that we're looking at is somehow some degree of genetically admixed. And we've gotten to the point where there are perspective papers out there, one from Mosaic and colleagues, noting that what we thought about homogeneity in the genetic ancestry within populations, it's an idealized concept that doesn't really exist. And again, I'll unpack this a little bit further. Before I dive in a little further, I want to make the point that when we think about genetic ancestry, that's a distinct concept from the self-reported socioeconomic or social determinants of race and ethnicity. There are many different papers that have been reported this, and here I'm just highlighting just one of them that appeared in the New England Journal a couple of years ago, that really makes the point that what people self-identify is distinct from the history of one's genome. And there's no other populations to exemplify this than admixed individuals, where genetic ancestry varies across the genome and varies individual to individual. The other big punchline here, which is, I'm sure all of us are fully aware of this, is that such individuals that have mixed ancestry are vastly underrepresented in genomics. Here's listed one of this plot where on the x-axis you see years going all the way back to 2000s, and on the y-scale you see the percentage of individuals in genomic studies that are essentially from various ancestry populations. And as we move forward, when we get more diverse ancestries, it remains the case that individuals of admixed ancestry continue to be underrepresented, partly because there's such a misconception that the statistics, or we don't have the methodologies to analyze and correct for ancestry properly. And if there's one take-home message that I have for you today, is that that's wrong. We do have quite a sophisticated statistical tool set to analyze data from admixed individuals. And just like a fun trivia, roughly 1/3 or 1 in 3 people within the US self-report as having admixed mixed ancestries, whereas in the genomic studies, it's roughly, 1 in 20. So you see like this massive underrepresentation. And according to the US Census, that number is going to go to around 50% by 2050. So for all of you that are more on the junior side, within your career, you'll have, if you work in admixed populations, roughly 1 in 2 individuals will be impacted by your research. Okay, so in order to kind of like dissect and unpack some of the methodologies here, let me go into how we traditionally model admixed populations. So in the most simplistic form, we think about it as 2 ancestrally diverged populations, let's say the red and the blue population there, that have been diverged for a while, and then people from those ancestries come and have offspring in a different environment and a different population. And due to recombination events, the genome of the admixed individuals starts recombining between segments from the different ancestral sources, such that the genome of admixed individuals have this unique features that they're a mosaic of segments of ancestry from, in this case, the red and the blue population. And the length and the distribution of this ancestry segment is another molecular clock that we can use to investigate the properties of this demographic event. Okay? But I'm not going to be talking about that today. Today I'm going to talk about how do we use that in genetic or in studying the genetics of disease. So that type of mosaic of genetic or segments of different ancestral sources induces the unique feature for admixed individuals where every individual has its own unique mosaic of ancestries, which leads to unique proportion in genome-wide ancestry. So the proportion of how much of one's genome comes from the red or the blue population in that case. And here on the left side, as you're looking, you see the standard way that people plot genetic data using principal component analysis, where individuals with mixed ancestry form a line from the 2 ancestral sources. Here on the bottom here, you see individuals that self-report as African American, forming a line from this an African genetic ancestry cluster towards a European genetic ancestry cluster. The same type of things or variation across individuals, you can see it on the right side, but there are various populations within Central and South America, where you do see every individual has its own unique proportion of African, European, or Native American ancestry. So every unique is personalized, if you will, based on this proportion. The fact that the segments of ancestry come on different, or the allele come on different ancestry segments, allows us to think about how do we consider disease mapping in admixed populations. So the first one is we need to correct always for genome-wide ancestry, because genome-wide ancestry is correlated with social determinants of health. So by correcting for it, we can remove that potential bias from our studies. And really the most interesting point here is that every allele, every potential causal allele appears on multiple backgrounds in populations of admixed ancestry. Here I'm showing you an example of an individual that is admixed European African, and you see an allele that's potentially causal having an effect size on the African background or red blue background here or a red background. Okay? So the fact that these alleles appear on different backgrounds allows us to ask questions whether these effects by ancestry are distinct or not in these populations. So in the next 3 slides, I'll try to disentangle one of the concepts that is often misunderstood in this area, and that's the concept of heterogeneity by ancestry. So how do we measure this distinction? How do we measure differences in the genetic effect across ancestries? The first one is the simplest one. The causal allele could have different impacts on disease as if it comes on, let's say, red or blue ancestry. So the effect size could be different because interactions exist, G by G, G by E, and so on and so forth. Right? So that's the simplest way to think about heterogeneity by ancestry. The other one is, even when the causal effects are the same, even when biology is all the same in every cell, right, because in the body, it could still be the case that these mutations have different frequencies in the 2 populations. So the fact that the frequency of this causal allele has vary across population induces a difference in the variant explained by these mutations in the disease status across populations, which is usually leads to an admixture signal, and I'll get back to it in a few slides. But that's also being referred as heterogeneity by ancestry, because now you can explain different proportions of the variants in the disease, even though biology is all the same, and the mutations have the exact same effect by ancestry. And finally, which kind of encompasses a lot of the challenges here, we often don't know the causals, and we're looking at something that it's tagging the causal, and therefore we have this taggability issue, and tagging varies, or it's ancestry specific. So every potential causal allele gets tagged differentially, whether it's on, let's say, European versus African background. So every time we think about heterogeneity, we need to think about at least these 3 sources, which I'll try to unpack a little bit more. So how do we kind of combine these into statistical model to do disease mapping, let's say, in admixed populations? Well, the standard way to do it is, what has been referred to as genome-wide association studies, which I'm sure all of you are familiar with, so I'm not going to dive too much into it. But essentially, I'm going to present it in a case control setting, but you can think about it continuous as well. What GWAS is looking for association or a difference in the frequencies of a particular mutation, cases versus controls, people that have or don't have the disease, which is rather straightforwardly modeled by a likelihood ratio test, where we're simply testing for an effect size of a particular mutation or a particular variant in the causal versus the non-model. So I put it here as a likelihood ratio test, but obviously you can craft this in all sorts of different ways to run this association stat. And as I said before, it's critical for admixed population to model it as, or to always correct for the genome-wide ancestry. Because admixed populations have alleles that appear in multiple backgrounds, we can actually disentangle this, and we can ask whether or we can write a model in which the effect sizes are decoupled based on this local ancestry background. So it's equivalent to a meta-analysis, for those of you that are more interested in that area, where we ask whether the effect size on the European background and the effect size on the African background are uncoupled, and we assign them the same causal effects, and this allows us to run an association scan that is treating these 2 effect sizes in different components of the lineage. And because I'm assuming the same odds ratio or the same effect size, then it's still a 1 degree of freedom test that it's equivalent to running this test in Europeans and Africans and then meta-analyzing under fixed effect assumption. But because we can do that, then we can already start modeling differential effects. So we can, if we believe that the effect size might vary by ancestry, we can start modeling them as effect size being uncoupled, European versus African, on the 2 local ancestry backgrounds. And we can allow them to vary as we want, and that will give us 2 degree of freedom test. Or we can make them similar or more similar, and that will allow us to play the game between 1 degree to 2 degree freedom test. And as you might expect, that will give us a trade-off between power versus robustness. The local ancestry signal, you can think about it as if the disease-causing allele is higher frequency in one of the ancestries, in this case, let's say in the red ancestry, then individuals that carry that disease will have more of those alleles at the disease-causing locus. And that induces what is called an admixture signal, or a correlation between the average local ancestry in the cases versus either genome-wide or versus controls, depending how you set it up. And for those of you that are junior in the field, this was actually one of the first ways to do disease mapping even before GWAS. So it's kind of interesting that now we're in some ways we're getting back to it and trying to merge it. We can exactly follow the same kind of paradigm where we can write a likelihood ratio test where we have under the null, there's no effect for the local ancestry. Under the alternate, there's an effect for the local ancestry. And we can write, again, a 1 degree freedom test that would be essentially equivalent to running a regression between the phenotype and the local ancestry status in the case of the individuals. Because we uncouple these, now we can start doing something more exciting, or more fun, if you will. We can, because we uncouple the local ancestry in the first step, in the case control study, we can essentially add these two likelihoods together, or multiply them, or add the local likelihoods, and we can mathematically show that these are independent, and this could be a way where we can sum the effect of the association with the effect of the admixture, and we pay the penalty of 2 degree of freedom test, but again, now we're essentially gaining more insights into how do we do admixture or association spans in a mixed individual. The really interesting bit that if you start writing down with math there is that you can actually, under some very simple assumptions, you can write down the induced effect on the local ancestry if you know the effect size of the causal alleles. And with some very straightforward manipulations, you can show that the ancestry effect size is a function of the allelic effects of the causal SNP and the frequency. And it has this very nice and simple formula there. And you can clearly see that if the SNP is not causal, so R is equal to 1 there, then the ancestry effect size will be essentially 1. Or if you see that the allele frequencies are the same in the two ancestries, again, it will cancel out irrespective of the causal effect. So this gives you insights of when should you expect a local ancestry deviation. It's either you need both effect size as well as difference in the frequencies. Okay, so I'll show you. I showed you a bunch of squares, and there's some math behind it which I didn't show you, but we can talk about it. How do all these tests operate in practice? And here are a bunch of simulations based on admixed individuals in the UK Biobank. So these are admixed individuals, African and Europeans. The simulations are power simulations under some reasonable effect size, which I forgot what it was, but the power simulations, I distinguish them between, on the left side is what happens at a randomly picked variant in the genome, and on the right side, what happens at variants that happen to have different allele frequencies between the two populations, with the idea that those with allele frequency difference are somehow more biologically interesting to look at. So with the methods that are compared here are just a flavor of the ones that I showed you before. The top one is just the vanilla GWAS with the correction for genome-wide ancestry. Step one is the one where you partition by local ancestry. This is just combining them. Tractor is a recently proposed approach that allows for a lot of heterogeneity by ancestry, by Elizabeth Atkinson et al. And again, all tests correct for genome-wide ancestry. So here's the most powerful thing that you can see in the simulations. If you just compare, these methods that do not adjust for local ancestry versus methods that adjust for local ancestry, you can already see that, in fact, per individual sample size, at the same sample size of GWAS, you get more power by looking at mixed populations than by looking in homogeneous populations. Because you get more signal between the differentiation. Because on the average, every SNP is slightly differentiated. So per unit, per individual that you add to a GWAS, you're more powerful to detect disease-causing associations in mixed populations. The other part is, as you might expect, if you condition out the effect of local ancestry, you're actually removing out some of the effect, and then you might lose some power. The positive is that you might be able to adjust for different heterogeneity by ancestry. And finally, if you're really going for lots of differences by ancestry, you reduce power by around 10% or so, but you're gaining in robustness because some regions might truly have heterogeneity by ancestry. Those are simulations. Here's what happened in real results. Essentially, you get slightly 10% less power by methods that account for heterogeneity across the board. And this is fully expected because you need to pay a penalty because of the extra degree of freedom test. But really, if you focus on just loci where you see a large frequency differentiation, and this is the case for this Duffy SNP, which happens to be one of the highest differentiated SNP between Europeans and Africans, you see these methods that correct for local ancestry completely miss this signal. And this is a well-known causing allele that causes a condition that it's been labeled by clinicians as benign ethnic neutropenia, which in non-medical terms means that the neutrophil counts is one standard deviation lower than the expectation, but it doesn't cause anything. So that's why it's the benign. So clinicians know that that's what's happening. This mutation causes this. And you see that by conditioning local ancestry, you completely miss out the signal, which is what you would expect, simply because local ancestry tags perfectly the association signal. Okay, so hopefully I convinced you that we have a lot of statistical methods to do mapping in admixed populations. There's many more. I'm happy to talk more about you, but the fundamental question here that you might have is, how do we select among these methods? What is our guiding principle? And what's fundamental there is whether we believe that the causal effects are the same or not across ancestry. And I pose this as, this is probably fundamental not just for admixed individuals, but also as we think about transferring genetic finding from, let's say, Europeans to Asians to worldwide population. Okay? And the unique features of different segments of local ancestry allows us, for admixed individuals, to actually ask this question in a very, very controlled environment. So within a given cell, all the cellular context is all the same for a particular individual, right? So we can study the effect of these alleles on disease without confounders from the environment. And we wanted to utilize this unique feature of individuals of mixed ancestries to try to tackle this question. Are the effect size on European backgrounds the same thing as on African backgrounds? And because we found out for the past 10, 15 years that all the human diseases have lots and lots of causal variants or risk associated regions, we shifted it from looking one variant at a time towards a genome-wide model that simply says, if I plot the effect size in Europeans versus the effect size in Africans, is the slope close to 1 or not? And is the variants explain 1 essentially. So is an effect size in Europeans predictive really well in Africans or not? And you can label that as a correlation in the causal effect, or a genetic correlation across ancestries, if you will. But it's really just saying whether the effect sizes are the same or not. And the way we solve this problem is through a mixed model approach, in which we leverage the fact that all human diseases are highly polygenic, and we're literally just figuring out a genetic relationship matrix between pairs of individuals that is restricted to the European segments, that restricted to the African segments, and we fit that through a model that's listed here on the right that makes an assumption that the effect sizes are distinct across population and they're allowed to be as distinct as possible. And we fit these parameters, which would be the covariance between the causal effects, which is this rho G here on the right side, versus the sigma square G, which would be the variance or the heritability in the two populations. And under this model, this quantity, this covariance divided by the variance there, this correlation, R admixed, we labeled it, will tell us whether the effect size are similar or not. So if this number is 1, essentially the genetic effects are all the same. If this correlation is 0, then essentially they're completely uncoupled. Okay? Hopefully that makes sense. And I should say that should interrupt me if you have any questions along the way or not. So as with all mathematic models, first we run a lot of simulations, and I'll guide you to the preprint that's listed there. to see all the simulations that we've done, but they are encapsulated in this figure on the left side. Pretty much, we observe that this method correctly recovers the genetic correlation, albeit is downward biased, mainly because if we mask all the true causals and we try to use the tagging. And the downward bias is small, but it's consistently downward and not upward. So all that motivates us to think that this is a lower bound of the actual genetic correlation in real life. Virtually any simulations that we've done or any breaking of the model leads to downward bias, not to upward bias. So you see here that we simulate from 0.9 to 1, and you see the estimator is very close to that ratio. So then we applied lots of data, and then here we kind of tried to look at all the data that we could get our hands on. Again, guided by the fact that sample size matters in this case, and guided by the fact that it's very hard to look at individuals of African European admixture. So we looked at the PAGE study, which is a multi-ancestry cohort. We looked at the UK Biobank. We looked at the All of Us, which overall gives us a sample size of around 50,000 African European admixed individuals. And that includes African Americans, that includes Afro-Caribbean from UK Biobank, that includes all sorts of individuals that are self-report in various different ways. So, long story short, what we found is that the correlation is very, very high. So it's on the order of 0.95. You can see the credible intervals there. On the left side here, you see a selected 10 traits that happen to have the largest sample size. And you can see that, the credible interval or the credible range for our estimate varies widely across trait because of the sample size, because of number of cases by that trait. But in a meta-analysis, it's very close to 1. And I'll guide you on the right side here, where we compared what would happen if we look at across continents. So here's if you look at African versus European, you get something on the wrong order, like a correlation of 0.5, 0.6. If you look at African or European versus Asian, you get something around 0.8. And if you look at the mixed individuals, you get something that is 0.95, which is what's listed up there. So this is very consistent with this idea that by looking or by disentangling by local ancestry within the same individuals, it's more robust to this confounders by environment. And in many ways, it's kind of eliminating as a local genetic interactions as a source for differential effect by ancestries. It's also consistent with the fact that maybe environment contributes more when you look at across continents than when you look. So there's G by E that may contribute to looking at transcontinental genetic ancestries. So that happens with that method, that global method. We confirm this by just focusing only on the genome-wide significance SNP, so that's what's showing you here. Now every dot here is just one genome-wide significance SNP. There's less power for this type of approach, but the number is very similar to what we got through our polygenic method. It's still higher than 90%. So the answer that we kind of convinced ourselves after lots of work within the past years that genetic causal effects seem to be the same across ancestry. So they seem to be very, very similar, and really what may be contributing to the fact that we see lack of transferability across population is frequency and maybe tagging of unknown causal variants. But once we narrow down on the causal variant, they seem to be operating the same across ancestries. That's kind of our conclusion. So if you kind of heard my last statement, there's some inconsistency there. So how can just different frequencies contribute to such a low transferability of polygenic risk scores across ancestries? And we started to try to unpack that a little bit, and I'll show you how that could occur. So I don't want to spend too much time on it. Probably all of you guys have heard about polygenic risk scores. They're hyped to be the next best thing, the next sliced bread, I guess, in personalized medicine. But essentially, it's saying that in GWAS, we found a lot of small effects, and none of these small effects can predict individuals that will get disease. But if we aggregate them through some linear combination, we've seen non-linear combination as well, but if we aggregate them up, we might be able to predict which individuals might be at the tail of the distribution, or which individuals might have higher genetic risk for some diseases, and we might be able to either, do prevention or personalize their treatment decisions, or in general, change our medication or change our clinical decision making based on their inherited genetic risk. And like I was saying before, this is probably one of the biggest challenges in making polygenic risk scores applicable in the clinic, is the fact that they seem to work much poorer the further away you're from the European ancestry. This figure that has been, probably one of the most consequential figure in the past, 4 or 5 years. You've seen it probably everywhere from Alicia Martin and colleagues. It's showing the predictive accuracy in polygenic risk scores, all refer back to the European ancestry training data. So you see here on the first column, accuracy of 100% in European ancestry individuals. Then if you move to, I'm going to use quote marks, American individuals, then you see the predictions are around 50%, South Asian, East Asian, African, and so on and so forth. Right? And now to unpack why I put the quote mark, it's unclear what American means here. It's like, are these folks that are coming from the US? Are these folks that self-identify as American? There's a lot of admixture happening in various different forms. And note that there's huge error bars there. For some American individuals, it might be 0.9 accuracy of when Europeans. For some American individuals might be 0.5 accuracy. So we try to unpack this spread of performance with the idea that maybe admixture is driving a lot of the signal. And we're not new to this. This is work from Jan Matthiessen and many others that are trying to look at the transferability of polygenic risk scores directly in admixed individuals. And I apologize that they shift the scale, so now you have to look on the right side for like higher European ancestry versus on the left side, which is lower European ancestry. And again, the Y scale is accuracy. And what they plotted here, they looked at multiple African American or African, some of them are African American, but some of them are African European admixed individuals from UK Biobank. And they plotted here on the x-axis the average European ancestry proportion. So higher on the right, lower on the left. And you see how individuals that have less and less European ancestry in their genome, which will be on the left side, they have lower and lower accuracy in the polygenic risk. Right? So you see how this effect is not just a population-wide, but it's from individual to individual. Right? So individuals that have more African, they have less accuracy. Okay?
Can I ask, does this polygenic risk scores for what? For diabetes, for cancer, for
in this case, it's height. So it's kind of the fact that they call it polygenic risk score. It's a little bit problematic, but it's a lot of the. That's why I tend to use polygenic PGS as opposed to PRS. But I should say that the statements extend to most common diseases in humans. So if you do the same thing for coronary artery disease or lipids or whatever or cancers, you would see the exact same trends. And because you asked that question, the error bars here, I think the error bars are across traits, not across individuals. So essentially in this plot, they looked at a couple of hundred or 80 or to 100 traits, and what they plotted the bars here as the standard deviation across those traits. Any questions? All right. So now I should introduce to you the biobank that we're kind of applied this data, and this is for those of you that have never been to LA. This is a very colorful map of LA that we use. UCLA is somewhere there in the blue shaded version there. Here it's the color coded is based on the self-reported race ethnicity, and I don't know exactly how they actually color coded there, but it's presumably as a function of which one is the majority in that neighborhood. And the punchline here is that it's very colorful. UCLA or LA it's one of those cities where there's no majority. There's the largest population of European ancestry, so you're like 40 something percent. and Hispanic another 40%, and then a lot of other diversity. Here on the right side, I show you a principal component plot of the first 40,000 genotype data from the biobank at UCLA. So this is literally patients that come to the clinic, they get biobank, they get convinced to participate in this study called Atlas, very similar to how the Michigan Biobank gets described here. And it's color-coded based on genetic ancestry, not based on self-report. But the point that I'm making here is that it's a very colorful, and hopefully some of the colorfulness kind of matches what's here. So we see diversity in our biobank throughout. And I'll focus more on this in the next couple of slides. The message that we are kind of beginning to emerge from all the biobanks. Here, this is the same plot that I showed you before at UCLA, so in Los Angeles. This is the plot from a recent paper in Science by Lewis et al. That shows in a way more refined color scheme than we did, because it's in Science. They show what happens at BioMe in the biobank at Mount Sinai. And what you kind of like take home from this is that the fact that we assign clusters, I don't know why this is moving, the fact that we assign clusters is very suboptimal. It's clearly, you can see that individuals kind of like span the entire range from, let's say, the European genetic ancestry cluster all the way to African cluster and so on and so forth. So you see individuals happening, kind of like occupying all sorts of space in this continuum map. And that motivates us for, trying to disentangle or try to see how do we incorporate this continuous way of looking at ancestry into polygenic risk pool. Okay? And just to unpack this a little bit more, what happens in the figures that I showed you before is that somebody, ran this PCA plot as before, then drew some circles around it, called these individuals as, you're African, East Asian, and so on and so forth. They assign these labels of these clusters. And then within every cluster, they computed some single metric of accuracy, and that's what you saw plotted before. Okay? So you see European, Hispanic, Latino Americans, so on and so forth. Right? So that's kind of like what happens under the hood every time that you see one of these, polygenic risk scores decay, or performance decaying as further you move away from Europeans. I'll try to argue in the next couple of slides. Yes.
So what does accuracy mean on my right-hand side exactly?
Accuracy in this case means the R square between the polygenic risk score and the trade value in a test held out data.
When you calculate the polygenic risk score, do you scale it once and use the same scale across all your samples, or do you rescale it in every subgroup?
That's a great question, which if you give me 10 slides, I'll get back to it exactly. But for this one, they don't rescale it because they compute this R square in each of the population by itself. The rescaling, I'm going to argue that we need to do a rescaling. Okay, so this clustering way to think about the impact of genetic ancestry into polygenic risk score prediction comes with at least 3 caveats that I'm going to try to convince you that, and probably you already kind of hinted towards them. So the first one is that by assigning a single number to an entire population, ignore the fact that there's variability across individuals. I don't know why this is moving. So by assigning a single number to, let's say, all the African Americans, you ignore the fact that individuals that have more or less African ancestry, then they'll have different expected accuracies. The second one is the arbitrary boundary, and the third one is there's a lot of people that are left out from these clustering methods. Okay, so let me dive in a little bit into the inter-individual variable. So if I take the cluster of East Asian American at UCLA, and then I run another principal components just on those individual labeled as blue there, then you start seeing more interesting clusters. So you see individuals that are of Filipino ancestry, individual of Korean, individual of Japanese. It seems very silly to assign a single accuracy value to all of them while ignoring the fact that these individuals are really forming different populations. Okay? So there's variability within every clusters. The other one is, like I said, assigning a single value for, let's say, all admixed individuals, in this case, let's say African American, kind of makes this very unreasonable guesses where you take two individuals that just happen to be on the one side or the other of the boundaries, and you would assign them totally different accuracies. So you might have an individual that you might think that it's, has an accuracy in R squared of 0.2 versus 0.4, because you assign them to different clusters, and that seems very unusual and problematic, given that these individuals are very similar in terms of their genetic ancestries. And finally, there's a lot of individuals that are left unclassified by all these clustering techniques. And that was a question that, I don't know when this was moving forward, but that was a question that some of you asked me over lunch. We actually looked more thoroughly, and more individuals over time get to be so complex in terms of genetic ancestries that all these clustering methods just fail to produce everything. So if you see in Atlas, after we did our best, we have around 6%, in UK Biobank around 9%, and Biome around 4%. And there's reason to believe that these numbers will keep on grow up over time because individuals will have more and more complex, they'll move to different places and genetic ancestry will continue to shuffle. Okay? So our idea is to try to incorporate this, what we call genetic ancestry continuum, and to come up with an individual-based measure of accuracy. Okay? So our idea is to take the clustering method approach with this labeled individuals and just simply, again, I don't know where it's moving, but just move it to a way where we can assign a distance for a given individual as farther away from the training data. And in terms of distance here, we look at the Euclidean distance in PCA space, but believe me that we tried a bunch of things. All the things that I'm going to say today are kind of insensitive to the choice of the distance. But there's very interesting things about how you weight these PCs. Okay, so after you get the distance, instead of having these like clumps of single number per populations, you can actually estimate an individual form of accuracy for a polygenic risk score, and hopefully that individual metric will go down across distance. Okay? How do we go about estimating this? I don't know why the slides keep on moving. But essentially, you can write down the mathematics of what will be the correlation between the polygenic risk score and the genetic value. So the genetic value is the estimate of interest for a polygenic risk score. And you can write it down essentially under some Bayesian approach. It has that form. Again, you can read more about it in a preprint that we put out on how you actually go about estimating it. But essentially, you can take the posterior of the variance of the genetic value divided by the heritability, essentially, and that gives you an estimated measure of R squared per every individual. Okay? The posterior genetic value, it's estimated by sampling from a method that gives you a polygenic risk score, such as LD-PRED2. Again, I'm happy to dive into the details of it. It gets very complicated very fast in this space, so I'm happy to write the equation for you and try to derive them. In terms of genetic variance, we're approximating it with the heritability of the trait, and that's an approximation. All right, so how does it look in real data? So this is real genotype data from UK Biobank, where we simulate data in the white British individuals. That's on the left side over there on top. And then we estimate this quantity as we look at individuals of different European ancestries and more within UK Biobank. So UK Biobank is a collection of 500,000 individuals collected within the UK. And after they collected, they reported where they're coming from, although they're living in the UK. So we see there that accuracy begins to drop quite linearly as the genetic distance kind of moves these people away from the training data, which is labeled as white British. Okay? And you see that, for example, even if you move away in individuals that are from Italy or Poland or let's say India, so you see that there's a decay in the accuracy that it happens to be linear. Okay? Now this is an estimate, so we can actually validate this by just binning individuals and then computing this R squared between simulated trait and the actual the PGS value, and we see exactly the same thing. So we see that our approximation works fine. And moreover, we have lots of other results that kind of convince us that by approximating this accuracy, you get a pretty good result. The really interesting bit happens when we actually look at real data. So here's real data in the Atlas Biobank. I'm plotting this relationship between genetic distance on the x-axis and the individual level polygenic, expected polygenic accuracy. So every dot there is one individual. And I plotted that in all these different populations, or with the idea that maybe we can see whether there are populations where genetic distance does not impact accuracy. And to a first approximation, there's no such population. So genetic distance impacts accuracy in all populations. So there's nothing really special about admixed individuals. We're all admixed in some way, or we all have some distance from the training data, right? That's a qualitative. On the quantitative sense, it is true that if you focus on admixed individuals like Hispanic, Latino, and African American, the correlation is closer to -1, versus if you look at more homogeneous population where the correlation is not as high, right? But that's a secondary factor. So just focusing on European American that are labeled as homogeneous, we do see from the two deciles, the closest and the furthest, that there's a roughly 6% decrease. So individuals that are further away from the UK white British, there's like a 6% decrease. We see a much bigger, if you focus on admixed individuals, which is what you'd expect, because genetic ancestry has much wider variability, up to, essentially the closest have an accuracy that's similar to Europeans, while the furthest have an accuracy that is roughly half. And in fact, if we just plot it in the same scale, in the blue are European American individuals within our biobank, and the same color is Hispanic Latino, we see that the top or the closest have the same accuracy as the furthest. And finally, just to kind of orient you about what happens if we look at all the data, we see that there's way more variability across individuals than across population labels. And that's what I'm trying to show you in this plot. So this is what happens in Europeans, this is what happens in Hispanic Latino, this is what happens in South Asians, East Asians, and African Americans. So you see a consistent linear decrease in the accuracy as you move further away from the training data where these polygenic risk scores were created. Most of that stuff that I showed you before is one example trait, but really we try to look at all the traits that we could put our hands on. Some of them are listed here. And to a first approximation, there's no trait where this does not hold. So this is kind of answering your question from before. The lowest correlation that we will observe for any trait was -0.86 across 80 or so traits, spanning all sorts of different markers. And that also holds across biobanks. So that's UK Biobank. This is an Atlas at UCLA. And then even if you look at just the European Americans in Atlas, there's still a much higher correlation. Okay, so that's all about accuracy. The second question is, and the final question is whether the polygenic risk scores themselves actually vary with genetic distance. So far I showed you how accuracy kind of goes down the further you move away. But how about the polygenic risk scores themselves? And I caution you that the polygenic risk score is a measure of genetic risk or measure of genetic value. So it could go down or up as a function of maybe the genetics of that particular trait varies with ancestry, or it could be a biased estimator of genetic risk in the data. So what we found is that all the PGSs, 82 out of 84, so virtually all, actually have a significant correlation with the genetic distance. So all the polygenic risk scores vary themselves across ancestry. But unlike the accuracy, the magnitude and sign, it's all over the place. Okay, so for some traits it goes up, for some traits it goes down, and that's kind of a reflection of maybe true signal, so selection changing this, or most likely some biased inference in the genetic predictors. And I'm going to exemplify this with just two example traits, and then I'll finish. One is about height, where we see on the top, this is the polygenic risk score, just in Europeans, going down just in the according to the distance in the training data. So you see how it goes down, the polygenic risk score there, and that matches really well with that, with actually what we see in measured height. So this is the average height in this individual's bent across genetic distance, and some people have used this as a sign of natural selection. Maybe genetics drives the difference north to south Europe, or other works have reasoned that this is an artifact of incomplete accounting for population structure when estimating these weights for the polygenic risk. This is the neutrophil count that I showed you before, and what you see here is that neutrophil polygenic risk score goes up at some genetic distance, while the measured difference actually goes down in some distance. Right? And this is again probably probably indicating this encoding of this Duffy SNP that has such an oversized role in the neutrophil. So the point that I'm trying to make here is that we should, for every time that we use any polygenic risk score for any disease, we should plot it across this continuum of genetic distance and reason about what could impact these correlations before we actually use it in diverse individuals. So in conclusions, I covered lots of stuff, and I kind of like us. I didn't go into lots of details on. Please reach out if you have any comments, or we want to go into specific ones. Hopefully, I convinced you that we have lots of methods for mixed individuals, so we don't want to remove any of those individuals from all the studies. All the methods that I showed you before are actually, we put them in this admixed kit, anywhere from mapping all the way to polygenic risk score. There's also, very nice software package from Elizabeth Atkins and others like Tracture. So we have lots of methodologies for mixed individuals. Hopefully I convinced you that causal effects are similar across ancestries. So to the first approximation, we believe that difference in frequencies are really the culprit for why polygenic risk scores, decay in performance across ancestries. I'm a firm believer that anytime that we look at any polygenic risk score towards the clinic, we should first interpret it through the lens of genetic ancestry in a continuous fashion. And by looking at individual level, it probably gives us a different look towards how we can identify subsets of patients, subsets of individuals for which polygenic risk scores work great, and we can use them in practice. So I'll stop here by acknowledging a lot of the folks that created the UCLA Biobank. Here's just, believe it or not, this is just part of them. And those of you that created the biobank here at Michigan probably recognize how big of an endeavor it is to create a biobank within the medical system. It's like just part of them, and ultimately acknowledge all the people that have done most of the work that I've showed you before. Them at SAG. Hopefully we interact with some of them. All right, so I'll stop here, and hopefully you guys have questions.
I was going to start and see if some students had questions they can jump in before the faculty. So, after being bold, particularly in training camp. Anybody, really?
I think whoever asks a question gets one of these squares. Yeah, go ahead.
So when you're evaluating the R squared by genetic distance, were you evaluating the R squared in in all of the sample together instead of the different populations, like how you said they were comparing R squared in the different populations?
Yeah, so we're looking at there's two ways that we did it. So one is by individual. So we have an estimate, essentially, that says for all the individuals at this genetic distance, this is what we estimate the R square would be. So if you sample an infinite amount of individuals just for that genetic distance, that would be the estimate that our method predicts it will be. And then to validate that, we bin the individual across genetic distance. And at that bin, we actually computed in a test held out data what we observed to be R square between the PGS and the trait value. And that's how we validated that everything lines up.
Questions? Open up the faculty.
The tough questions can come.
Gary?
there any reason to think it has to be a one-dimensional metric? You've mapped for PCA1 and PCA2, or maybe it was 10 PCAs, I don't know, and to one dimension, but you can imagine a two-dimensional space with distance there and sort of interpreting things in that space.
Yes, you're absolutely right. And we did this just for convenience, just as a first step in it. So we looked at the top 20 PCs and we look at the Euclidean distance across them, so it gives a single number. But you're absolutely right that there's no reason to just look at one dimension. And we hope that in the future we might be able to look at not just the PCs, but other ways where we can, we can come up with measures in multiple dimensions that give us a continuous reasoning of how one individual is further away from the training data that the PGS was creating. Yeah, that's a terrific suggestion that kind of probably motivates some of our future work or somebody else's future work as well. And I should say that probably goes really well with maybe looking at other factors that we typically add when we predict risk, such as environmental and other types of variables that we typically add when we take the PGS to actually predict relative or absolute risk for patients. Yes?
So I don't work in genetics, so I hope this isn't a silly question, but the polygenic risk score weights the SNPs, and the assumption is that the weights are the same across the races. But if there are interactions with genetic factors, with environmental factors, then presumably that wouldn't be the case. So is that a reasonable assumption, if it's true?
Yes. So the basic assumption of polygenic risk scores is that once you dive to the causal variant, the weight that's being used is the same when you're going to employ it across different ancestry groupings. But then there are ways, because we see that the polygenic risk scores in various different ancestry vary across different genetic distances. We can rescale the predictions of the polygenic risk scores, which essentially would mean to readjust the weight, because everything's linear, we need to readjust every weight that we use in the model according to this genetic distance. So to account for the fact that
relative contributions would remain the same.
the relative control remains the same, yes.
Which might not be the case if there were.
Which might not be the case, yes. And there's in many ways, there's more work to be done about how do we model G by E, if that exists, for various traits in this sense, yes. Please.
I think Laura first.
Okay. So it's kind of related to Rod's question. So when you showed at the start, when you look at admixed individuals, your betas for the causal variants look pretty similar to each other. Within admixed individuals, depending on who you're choosing, like if you're choosing African American individuals, there still is most individuals are closer to 80%, right, admixed African to European. So the genetic background that they're feeling globally, when you think about interactions, is still pretty similar. So when you try to infer that they're, I think you talked about local, right? You can infer what the local thing is, but it doesn't actually say if there are different genetic backgrounds, right? More different than you get in admixed individuals that are influencing those effects.
Yeah, you're absolutely right. So most of the, I think the average genome-wide ancestry in African Americans in the US is around 0.8, plus minus a standard deviation of 0.12. So there's that the range is somewhere between, let's say, 60%, the average up to 100%. But you're absolutely right that the backgrounds that might exist in other ancestries might be wider variants, and that may allow for the environment to contribute more by G by E, or just different E that will correspond to different phenotyping that will introduce different weights. And in many ways, if I can, this is not cooperating that well today. It seems like it's going forward by itself. Let's see if I can make it go backwards. But I showed you, and I went super fast over it, my apologies, but oops. wants to do its own thing. But this is exactly what this plot is trying to show. So this is African European admixture, okay? And there's a bunch of populations there, but for a first approximation, you can consider what you said, 80% plus minus 12, right? This is African versus European. So this is people from Africa versus people from Europe. And applying a very similar model, right? Ignore the statistical part. But you see that the similarity with the causal effects is 0.95 here, and this is 0.6, 0.5, 0.6. So you see a massive difference between them. In many ways, this was kind of, to me, the most surprising finding in this work, which suggests that there's a lot of E or environment that either fluctuates between the Europeans and Africans, which exist, but that could indicate that there's a lot of G by E that exists. So that's one potential direction forward. Or it could be that the phenotype that we measure in these individuals could be itself be different. There's a lot of work that looked at mental health phenotypes, and those are measured totally different across continents, which will induce, essentially, we're looking at different traits in many ways. But that's consistent with exactly what you are saying, that there might be a lot of E that we need to take into account, particularly if you look at different continents.
I guess I was trying to actually say the opposite, that you don't know if everybody was 80% admixed,
Yep.
for argument's sake, right? Those European and those African alleles are feeling the same genetic background. So we pretend there's no difference in environment. Right now you take and you put them in somebody who is European, or you put them in someone who's African, they are feeling a different genetic background. So you don't really have a way to, even from what you've done, say that it's environment and not genetics. Right, because that also varies.
Yeah, but I think you're making the assumption that if you look at two individuals with 80%, that it's always the same 80%. That's never the case. So if you look at two individuals that have the same 80% African, the actual alleles that are summing up to be 80% are all over the genome. So there's a randomization that happens there. I would agree with your statement if you're assuming that two individuals with 80% always have the same, let's say, polygenic background, it's always the same 80% throughout the genome. But that's not really the case. But again, we can discuss and we can try to figure it out.
Right. So very quick. So the main conclusions, I think, all make sense to me. I'm curious about your suggestions move forward. So what you observe these two seemingly contradicting things. One is the causal variance. Well, the causal effects are pretty consistent across the population groups, and then the transferability of the PGS. So what should we do?
Yes, I think the transferability of PGS is directly addressable by differences in frequencies. So I think that would explain almost everything. I do think that in admixed individuals, the causal effects are almost always going to be the same. So we can, use that knowledge when we design. And by causal effect, I mean the allelic effect. The frequency was still there when we design, all sorts of methods from mapping to polygenic risk scores. And when we look at transcontinental or other types of non-admixed, if you will, individuals, then there's a role for a lot of environment to change the way we kind of observe these effects.
Right. I think a lot of reasoning say this is an environmental effect, but is that possible? We're focused on the GWAS findings mostly using the European individuals, so there's some risk. Alleles maybe is very rare in the European, but quite common. So the point is probably ask for your comment. Should we do more GWAS in other?
Yeah, of course.
It's like
groups.
I think it's just the overall set of the causal variants there.
Yes, yeah, you're absolutely right. So maybe I didn't emphasize that enough. Yes, there's all the variants that we're using in all these studies are variants that were observed, biasedly in Europeans, because in Europeans we have large sample size. there are a lot of variants that contribute to the disease that are, let's say, absent or low frequency in Europeans that may have high frequencies in non-Europeans. And, I can't make any statement, or I didn't make any statements about them because we haven't really observed them. So we definitely need, more studies in non-European for sure. And that's just on principle level, if you will. As you probably know, and all of you know that, by analyzing these populations together, we can, narrow down where the causal variants are more accurately, and we can essentially answer these questions about the existence of G by E or not. So in any means, I didn't want to make the point that that we should, study only homogeneous population. Quite the contrary. I think looking at as much diversity as possible kind of gives us this new insights. And by diversity here, I mean, genetic diversity in our studies.
So I'm a little bit confused. When you say that you expect that environmental differences make a big difference in transcontinental population, that seems to be in a contradiction with your very nice linear decay of, right?
Yes.
That is much more about what you would expect if it's really just differences in linkage disequilibrium and allele frequency.
Yeah. That's where I fully agree with you. That's where I think it's worthwhile to dissect this more. I think it's consistent with like a G by E, but I'm not fully convinced. Yeah, that it's a G by E, or it's just bad estimation. We don't have the context. Hopefully, we can tackle it.
Other questions?
I'm not a fact even.
No, you can still ask it. Everybody.
Yeah.
These are not mine to give, but
How do you think about this question when you model the causal effects or cross ancestries? Are you still focusing on a specific region or saying locus? You are trying to average over this locus rather than directly saying one causal SNP is causal for one SNP is causal for one ancestry and the other is causal for other ancestry. Is that an average proportion or is like for each causal variant? And with that, is that possible that we have potentially different causal variants for the mechanism across ancestries and therefore even though they have very similar causal effects across ancestries in terms of the total contribution of the heritability across ancestries, but there are still different causal mechanisms across different ancestries.
Yeah, that's so just I went super fast over this, and my apologies for not going into. So in all this work, we've fitted different effect sizes in the two populations. So we allow them to be as free as possible, and we did it in two ways. So in one way is to average across all the variants in the genome in a polygenic setting by saying that the similarity in the genetic relationship matrix should match the similarity in the effect sizes. So this is the standard Dremel type of approach, and that gives us that single number 0.95. And then we did it the other way, where we just looked at the top index variants from the GWAS before. That's maybe it's a slide before if this computer allows me to do it. So we did. This is where every dot here is one variant that comes from GWAS. And in this case, we did LD clumping to kind of, and we played with a bunch of different ways to do it in terms of how do we account for the fact that we don't have the causes. This is what you're saying. You're saying that these variants are not causal. We're looking at this attack variant by region, and if we have the causal, then we might see something different. So to a first approximation, we tried all the ways that we could look into it, and we always see this, which is that it's highly correlated, either by index, by fine mapping, by clumping. And I think this results, particularly in this plot, are LD clump. So we did LD clumping in kind of like ancestry specifically. And then your other question, I think it's we can still see different effect sizes by ancestry at particular regions, and you'll see here that there are some, let's say, especially look at those red ones there, where although overall it's very similar, there might be regions where it's truly the case that the effect size will vary by ancestry. And then finally, I think it touches a little bit your question about whether the the causal mechanism might be slightly different by ancestry. I don't know, it seems a little bit hard to say. As far as all the work that we've done, or we've seen, it seems like the causal mechanism seems to be more or less similar across ancestry. Albeit that, variants might have different frequencies and it might explain different amounts of this. Hopefully that addresses your question. One more?
Okay, let's
Thank you.